Hi, good morning, good afternoon, and good evening to everyone here in America and around the world. Welcome to today's session, the third session of the Sam Gambier Early Professionals Forum. And just a reiteration of Dr. Sam Gambier, who everyone here is quite well aware was a world-renowned clinical scientist and visionary in the field of molecular imaging and theranostics. And I apologize, I think I need to mute myself on the other one. Sorry about that. And so this forum was created in honor of Dr. Gambier and his unwavering dedication to mentorship of future generations of imaging scientists. And with that, for each session, we've invited senior scientists as well as new young professionals to introduce their research and their perspectives on mentorship and being mentees. And so today we have the honor and pleasure of having City of Hope with Dr. Anna Wu and Dr. Kirstin Zettlitz as speakers today. So first, I would like to introduce Dr. Anna Wu, who is professor and chair of the Department of Immunology and Theranostics, and is the co-director for the Center for Theranostic Studies at the Beckman Research Institute in the City of Hope. And she holds a secondary appointment in the Department of Radiation Oncology at the City of Hope. She was previously professor and vice chair at the Department of Molecular Medical Pharmacology at the David Geffen School of Medicine at UCLA, and was also director of the Cancer Molecular Imaging Program of their Cancer Center. Many of you know she's well experienced and expert in the field of ImmunoPET, and she is the co-founder of ImagineAb, which develops and commercializes radio-labeled antibodies for clinical use. Dr. Anna Wu received her bachelor's degree at Harvard University and her PhD at Yale University, and did her postdoctoral also at Yale University and UCSF. And without further ado, Anna, would you like to share your presentation?
Yes, thank you so much, Jason, and everyone for your the opportunity to present in this forum that both honors Sam, but also highlight some of our early stage professionals. Sam was always a big fan of young investigators and really wanted to help them bring their ideas to fruition. So I first have some disclosures, and as noted, the main one that's of relevance here is that I'm a board member and consultant to ImagineAb. And this talk today is going to be a science, technology, and utility talk, where I'm going to talk about antibodies and radionuclides and how we put them together into ImmunoPET and some applications. But it's also a personal story because my path in ImmunoPET is intertwined with my relationship with Sam. So I started my career, my independent research career at City of Hope, where I worked on engineered antibodies for radioimmunotherapy, radiopharmaceutical therapy. But I spent a sabbatical year and then was recruited to UCLA by Sam, where much of our work focused on ImmunoPET, along with other applications for of antibodies for molecular imaging. And along the way, as mentioned, I founded a company called ImagineAb. I'll tell you a little bit about that. But Sam was a big proponent. You can see he was a member of our original scientific advisory board. And then 4 years ago, I was recruited back to City of Hope to co-lead our clinical theranostics program. So that's the story I want to tell you, and you'll see throughout the impact that Sam has had, not just on early stage professionals, but also on mid-stage professionals such as myself. He was an inspirational leader and a wonderful colleague. So this is where it started a long, long time ago when I was at City of Hope. And I was working with a wonderful group of collaborators because it takes a team to do this kind of work. And in particular, Jack Shively had identified some really, really specific antibodies towards a key biomarker in colon cancer known as carcinoembryonic antigen. It's also expressed in other adenocarcinomas, as you can see in this example of an invasive breast cancer stained for CEA on the right. And his group had a wonderful high affinity, highly specific murine monoclonal antibody that had absolutely no biological activity. So the thought was, he and actually Dave Beatty, who's not in the photo, thought, let's take these antibodies and radio label them and use them to target to colon cancer in patients. And initially with indium-111 for SPECT imaging and then yttrium-90 for therapy. So here's a very early example of theranostics. And I joined the group a bit later, really to head up the antibody engineering group. I took it over from Art Riggs, where we advanced first a chimeric mouse human antibody. And then ultimately, we fully humanized the antibody to produce M5A, which is now the workhorse for our clinical trials targeting CEA in patients now. And also developed antibody fragments really, with the goal of optimizing antibodies for delivering radioisotopes in patients. So that's the beginnings of the group. And some of us are still at it today. For example, Jeff Wong and Jack Shively and myself are now, we got the band back together at City of Hope and are still working on these projects. And so this is the concept behind those engineered antibody fragments. The challenge with intact antibodies is that in their native form, they have long circulating half-lives of days to weeks. And if you put a radioisotope, a therapeutic radioisotope on, this would lead to bone marrow toxicity, since there'd be excessive exposure to the marrow from the circulating antibody, and the marrow is highly radiosensitive. So we approached this challenge by engineering antibody fragments that would be optimized for radioimmunotherapy. They would exhibit rapid tumor targeting and rapid blood clearance to spare the bone marrow. And in particular, we focused on mini-body, shown here in the middle, and larger fragments such as single-chain FVFC, because they should have hepatic clearance, not renal clearance. And that would also spare kidney toxicity by having fragments clear through the liver. And I was very fortunate to be at City of Hope because they had a very active interest in translating agents into clinical use. They had, while I was there, they built a CGMP manufacturing facility, which can also make proteins, biologicals for clinical use. And as a scientist, as a basic scientist trained in biochemistry, for me, it was such a thrill to be able to see our chimeric T8466 minibody labeled with either iodine-123 or indium-111 make it into clinical evaluation as an imaging agent in patients with colorectal cancer. But what about PET? So back then, trying to put together antibodies in PET was a real challenge, because pretty much the main radionuclide that was available for positron emission tomography was fluorine-18. And as you know, that has a half-life of 109 minutes, a physical half-life. So that was like the irresistible force. And then we try to combine it with what I thought of as an immovable object, which is an intact antibody. As mentioned earlier, these things take a long time to target and clear from the circulation. So by the time your antibody gets where it's going, your radioactivity, if you're using F-18, would be long gone. So this was the fundamental challenge in trying to bring forward ImmunoPET. And as you know, there have been advances both on the antibody side and the antimatter side. So the field has benefited tremendously by much broader availability of positron-emitting radionuclides with longer physical half-lives, such as copper-64, iodine-124, more recently zirconium-89, which has really revolutionized the field, in addition to gallium-68, fluorine-18, which were widely available, but more compatible with shorter, more rapidly targeting agents. And I will say that we did not invent ImmunoPET. In the early 90s, there were a number of pioneering investigators that were trying to put positron emitters onto antibodies with some success. These include people like Aga Apenitos, Steve Larson, Michael Zaletsky, Mike Welch and Sam Wells, and others who put either gallium-68 or fluorine-18 onto Fab or Fab prime 2 fragments. These are just enzymatically derived antibody fragments, or used copper-64 or I-124 in combination with intact antibodies. But as you could see, those combinations were not necessarily ideal. I think the other big challenge was you couldn't get these longer half-life radionuclides back then in the early 90s. You had to make them, hence I-124 were coming out of Memorial Sloan Kettering and copper-64 coming out of WashU. But we felt we had a solution to some of these issues, which was indeed these engineered antibody fragments, which we developed for therapy, but they actually also had ideal properties for imaging. So many bodies show peak uptake at 4 to 6 hours after injection in mouse models with a half-life of about 12 hours or so, and show hepatic clearance. The diabodies show even faster kinetics. They reach their peak uptake, as you can see here, in 1 to 2 hours after injection into mice anyway, with clearance half-lives of 3 to 4 hours. Renal clearance, because of their lower molecular weight, they're below the threshold for first pass renal clearance. And we thought these diabodies actually would be really good candidates for rapid molecular imaging agents. So along the way, I took a detour to UCLA. And this came about because at City of Hope, I met Patty Phelps. She was a researcher in the neurosciences division. She and I both lived in the San Fernando Valley, but we're working in the San Gabriel Valley. So we ended up carpooling for years. And her husband was Mike Phelps, and he was the chair of molecular medical pharmacology at UCLA and also the inventor of the PET scanner. And he led a very, very active program in developing PET radio tracers. He had just hired a brilliant young physician scientist, Sam Gambier, to start basically what was the molecular imaging program at UCLA. And Sam and Mike invited me to spend a sabbatical year with Sam, addressing some new areas in the field of molecular imaging. So Sam posed the question, what if we could directly image gene expression in vivo? The whole goal was to be able to do molecular imaging to look at very specific targets, but non-invasively in vivo, initially in mouse models. And so my task for my sabbatical year was twofold. One was to try to establish the baseline of could we directly image mRNA expression using radio-labeled antisense oligonucleotides, and initially we targeted HER2 with Chartrine Sun helping me with that work. But what I brought was this diabody that would target and clear within 1 to 4 hours. And the thought was, let's label it with fluorine 18 and see if we can do ImmunoPET. We didn't succeed my sabbatical year, although we worked at it with Joe Walsh and Tatsushi Toyokuni, and I'll give you an update on where that project ended up. But what we did do was put together the mini body, which had a slightly longer half-life, with copper-64. So this was a three-way collaboration amongst City of Hope, UCLA, and WashU to get this done. And it took, taking advantage of the wonderful prototype, the original micro-PET instrument built by Simon Cherry and his colleagues in the Crump Institute. And so this targeted CEA was labeled with copper-64. And what you can see on the right was we were able to get excellent targeting and imaging at 5 hours after injection in a mouse model where the mice had CEA positive colon cancer on one side and a negative tumor on the other side. And as you can see, liver clearance and low background in the body. And this localization by micro-PET was confirmed by digital whole body autoradiography, as you can see on the right. Again, you can see the really outstanding antigen-driven targeting in the positive tumor. So after sabbatical, I was very fortunate to be recruited on a permanent basis to the Crump Institute in Molecular and Medical Pharmacology. By this point, Sam was the director of the Crump Institute and was in the process of building a department there. And during the following years, while I was at UCLA, we did finally finish my sabbatical project with the help of Weibo Kai, Sean Chen, and Sam, where we took the anti-CEA antibody, actually Weibo did, radio-labeled it with fluorine 18, and was able to demonstrate in CEA positive versus CEA negative tumor, very nice imaging from 1 to 4 hours after injection. So that proves the principle that this was a viable combination. Along the way, during my sabbatical, I also probably did my last hands-on PCR reaction where I introduced the initial mutations into the diabody to add cysteine residues for site-specific conjugation and radio labeling. I made 3 different versions. The one we ended up with is shown here. This project was taken on by Tove Olofsson, but this also has been a mainstay of our follow-up work to be able to do site-specific conjugation and labeling as opposed to random, which is, I think, more important in these smaller proteins where you really want to control the location and stoichiometry of the conjugation and labeling. And a third thing that developed during these years was while I was on sabbatical, Sam introduced me to Rob Ryder, who was a professor in the Department of Urology at UCLA. And he and Owen Whitty had identified this target called prostate stem cell antigen, or PSCA. And Rob had, his group had developed this very nice mouse monoclonal antibody that recognized PSCA. And the thought was, well, maybe this is an interesting tumor target to go after, both for imaging and therapeutic purposes. So on the right, you can see a very early experiment that we did where we radio labeled his mouse monoclonal antibody with iodine-131. and injected into mice bearing prostate cancer tumors, sub-Q tumors that express PSCA. And in this case, we weren't doing PET imaging, we were just doing digital whole body autoradiography. But what you can see is physiological uptake in the stomach, which scavenges iodine, but you can also see very nice uptake into that PSCA positive tumor. And to make a long story short, Rob and I have continued to collaborate over the years. We humanized the antibody, we made mini-body, diabody fragments out of it. We went through an affinity maturation process, and we actually manufactured this affinity matured humanized anti-PSCA mini-body at City of Hope and radio-labeled it with iodine-124 for clinical imaging studies at UCLA. And here you see Rob and I with the investigational new drug application. They were paper filings at that time, and it was 2,000 pages. So to wrap up this part of ImmunoPET, also over the years, what we've shown is that this really is a platform that you can take any antibody with desired specificity. So here are a bunch of interesting tumor specific or tumor associated antigens in oncology. And you can take that intact antibody, humanize it, reformat it either into a smaller diabody fragment shown on the left or a mini body as shown on the right. You can radio-label them. It's a platform. We've done fluorine 18, copper 64, zirconium 89, iodine 124, and these will all give you ImmunoPET agents that can be used to image typically 18 to 20 hours after injection for the mini bodies, but as early as 1 to 4 hours for diabodies. And what I do want to point out here is the students and postdocs who actually did all of this work, really a very talented and hardworking group. They're the ones that did all the work. I just get to show the results of their labors. So I'd like to actually thank all of them, at this point for their contributions. Okay, and so what about imaging immune responses? This has been where my work also pivoted, once shortly after I got to UCLA, because it became apparent that there are a lot of additional interesting questions we could answer by imaging cells based on their cell surface phenotype. And particularly in immuno-oncology, it turns out that FDG-PET is not as specific as we would like when it comes to immunotherapy, because immune cells also upregulate their metabolism and become FDG avid. So it's difficult to tell whether the signal you're seeing is due to tumor growth or due to an active immune response. But we do have a wonderful set of cell surface markers, very well characterized cell surface markers of immune cells that define lineage, differentiation, and activation state, including checkpoint molecules and their inhibitors. And so this would open the window to many applications in trying to do non-invasive imaging to evaluate immune responses and inflammation and cancer immunotherapy. So we tackled this actually inspired by Tony Rebus, our melanoma immunotherapist at UCLA, who basically told us, that when you're looking at immunotherapies, many of them converge on the CD8 cytotoxic lymphocyte as the actual immune effector cell, as the active agent. They can be checkpoint inhibitor therapies, but they work to release CD8 T cells, vaccines, immunocytokines, bispecifics that recruit T cells to the tumor microenvironment, or cell-based therapies, many of which are CD8 positive. So our initial targets are shown here. I'll talk a little about CD8, but actually Dr. Zettlitz will tell you more about CD20. But it raised the question, can we image immune cells and can we image responses to immunotherapy using ImmunoPET? So this is work by Richard Tavares, our workhorse for imaging CD8 T cells in mice. It is a cis diabody, as I mentioned earlier, for site-specific conjugation and radio labeling, derived from a rat anti-mouse antibody. The parental antibody actually is depleting, but we don't want that biological activity, and that's one of the advantages of using one of these engineered fragments. They're biologically inert. And these are just two examples from Richard's paper showing in CT26 syngeneic colon cancers treated either with an anti-CD13741BB antibody or with an anti-PDL1 checkpoint inhibitor antibody, the difference in the T cells in the tumors, this is an untreated mouse versus a treated mouse. You can see the smaller tumor, the stronger signal, the activation of the full immune system. And in the checkpoint inhibitor setting, similar to people, not all mice respond. So both these mice were treated, but you can clearly see a difference in the non-responding mouse versus the responding mouse in terms of the distribution and intensity of the CD8 T cells in those tumors. And over the past few years, we've now done a variety of models and a variety of wonderful collaborations looking in graft versus host disease, checkpoint inhibitor therapy, adoptive cell transfer, combination therapies, oncolytic viral therapy, and using bispecific antibodies, all in preclinical models, and have been able to show the potential utility of directly monitoring that CD8 T cell response to these immunotherapies or these other disease settings. So what about clinical translation? So this is where we talk about translation and commercialization. And I'll say right up front that starting a company was never on my list of things to do. What I did learn in academia was that the wonderful facilities at City of Hope that enabled in-house production allowed a near basic scientist like myself see one of my ideas, make it all the way through into clinical translation. You know, really, to me, that was just a wonderful moment to be able to see these agents in patients. But I also learned that grant funding, you know, we had a program project grant, they could only support early phase trials, say 10 to 20 patients. And even if that 10 to 20 patients look good, the NIH grant structure and budgets simply cannot support phase 2 trials, or let alone phase 3 trials, whatever it would take to get things, you know, FDA approved and more broadly available. So what I learned along the way was that you need a commercial path. You need to find some sort of industry collaboration or funding if your ultimate goal is broad availability, translation. And the ultimate translation to me is to have something available so that a physician can write a prescription and use it. So I came around to the idea that maybe we need to do something about this. I learned that imaging is a tough sell. And nobody was interested in licensing this. So in the end, we realized we had to start our own company. And it continued to be a tough sell. We founded the company 15 years ago. But what I have learned, and this is really in a nutshell, those of you thinking about commercializing, make sure you have a good idea. Make sure you have intellectual property and patents on your good idea. Make sure you're addressing an unmet medical need, you know, because that's going to be the draw that is going to bring in other investors and people who understand that there is a need and a market out there for what you're developing. And I would say, above all, you have to have passion for what you're doing, and you have to have patience, because it is a long slog. But here are some snapshots of that slog. This is ImagineAb headquarters in Inglewood. This is our official ImagineAb Mini. Our first 2 employees, David Ho and Ari Lippman, setting up their interim lab bench until we actually were able to bring in benches. These are the 3 founders, Chris Barronbrook here with the forklift, and myself and Rob Ryder in the winter of 2007 in Boston, trying to raise money for the company. Here we are, I think about 2010 or 2011, the whole company. And of course, as I showed you earlier, Sam on our SAB. And from this tiny little shoot, we now have trevmiralimab, Berdoxan, which again, Dr. Zettlitz will be telling you about later. And I've come full circle now. I've moved back to City of Hope to focus on therapy, because in the end, that is what we want to do, treat patients. And delighted to be co-leading our clinical theranostics program and revisiting prostate stem cell antigen. We continue to work on that A11 minibody that I described briefly before, that I showed you the IND for, but it showed potential as a therapeutic agent. This is work by a grad student, Wenting Tsai, where she showed in a dose-dependent fashion increasing tumor growth inhibition by iodine 131 labeled A11 minibody. But it turns out this fragment was showing unexpected clearance to the kidney, which is what we don't want. We want to spare the more radiosensitive kidneys and direct clearance to the kidney. So this is our next generation agent, which Dr. Zettlitz actually has led the engineering and development of, showing that it's a larger fragment that makes it clear via the liver, but it has 2 mutations in the FC region that allow it to not to interact with the FcRn receptor, and instead exhibits much more rapid clearance than intact antibodies. So here at 24 hours, you can see it's basically cleared from the circulation, but shows excellent targeting to the antigen positive xenograft. And we're very excited that we just received an R01 grant that will take us all the way through into a first in human ImmunoPET study, first either copper 64 or possibly zirconium, and then ultimately into a therapy study. So I'll just summarize that ImmunoPET now is much more broadly used, and that's because many people recognize the power of combining the specificity offered by antibodies with the sensitivity, resolution, and quantitation offered by PET. And as I mentioned, the advances in antibody therapeutics means you can manufacture these. These longer physical half-life radionuclides have really opened up PET imaging to use of biologicals with longer half-lives. And we feel our contribution really has been to try to truly optimize these antibodies as imaging agents and therapeutic agents. So I'll stop there and thank my current lab at City of Hope. This is actually my lab, the new Zettlitz lab, and our small animal imaging core. My previous lab at UCLA, my many collaborators at the Frump and other faculty. But above all, to thank Sam Gambier, because he really inspired all of this work, and we are, continuing to push forward to realize our dream in his dream. Thank you.
Thank you so much, Anna, for a wonderful historical context of the ImmunoPET and of who's who in the field of ImmunoPET as well. Before we continue, just a quick mention that after the presentations, please feel free to enter your questions here, and they will be addressed at that point in the chat. And also at the end of the session, on your left-hand side, you'll see a link to the social lounge, where the speakers are inviting everyone to please join them, and feel free to have casual conversations and further questions with them on anything of interest. So our next speaker is an early young professional, Dr. Kirstin Zettlitz. Dr. Zettlitz received her bachelor's and master's in technical biology from the University of Stuttgart, Germany, and conducted her doctorate in the field of antibody engineering in Dr. Roland Konterman's lab at the Institute of Cell Biology and Immunology, also at the University of Stuttgart, Germany. From 2010 to 2016, she continued her postdoctoral training in the lab of Dr. Anna Wu at the Crump Institute for Molecular Imaging at UCLA, where she led efforts in the development of engineered antibodies recognizing PSCA, as you previously heard. In 2021 of December, she started her independent research program as assistant professor in the Department of Immunology and Theranostics at the Beckman Research Institute at the City of Hope. And her research focuses on antibody engineering and antibody fragment-based theranostics for the detection and treatment of cancer and inflammatory diseases. And so I invite Dr. Zettlitz to please share your presentation, and we look forward to hearing more about your work.
Thank you, Jason, for the nice introduction, and thank you, Anna Wu, for setting this up. I think it's quite obvious why we want to use ImmunoPET imaging to study immune cell subsets and what the medical need for that is. So for most of the immunotherapies that have shown quite some success in the clinic, it is clear that the CD8 T cells are the cytotoxic effector cells. So the idea that you would want to know if the CD8 T cells infiltrate a tumor or are in the tumor microenvironment makes sense, because this might be of a prognostic or predictive value. But then there are also other lymphocytes that more recently have come into focus, and people are interested in seeing, for example, tumor infiltrating B cells, because they can have a co-stimulatory function, they can act as local antigen presenting cells, and they can also make tumor-specific antibodies. So the co-localization of B and T cells in or around the tumor is associated with improved survival. And furthermore, if these immune cells come together, they can form lymph node-like structures that are called tertiary lymphoid structures. And here's an example shown in breast cancer, where you can see that they form active germinal centers with a B cell zone and then surrounding T cell zones, CD8 and CD4 T cells. And these tertiary lymphoid structures are associated with anti-tumor immunity. And so their presence in the tumor or the tumor microenvironment might be of a higher predictive and prognostic value. So the questions that we want to answer is, can we image these different immune cell subsets, and can the pre-treatment immune profile predict the response to immunotherapy and help us select the patients that are most likely to respond to therapy? And can we use the immune cell subset imaging both in the tumor and systemic as an integrated readout of response to therapy? And to do that, Anna has already introduced the CD8 imaging done by Richard Tavares, who used the CD8 specific diabody site specifically labeled with zirconium-89. And here I just show a wild type mouse where you can see the entire immune system. You can see all the lymph nodes and high uptake in the spleen. And then we've also developed that specifically for the CD4 T cell subset. This is work done by Amanda Fries. And similar, you can see the lymph nodes and the spleen, and then some clearance to the kidneys because of the size of the diabody. And then lastly, we use the mini-body targeting CD20 to image B cells. And again, you can see the lymph nodes. Because this is a mini-body, a larger fragment, you see some clearance to the liver and then high uptake in the spleen. And you can also appreciate that the ImmunoPET scans reflect very nicely the respective fraction of cells in these tissues that are of the different immune cell subsets. So B cells have a lower frequency in the lymph nodes compared to the spleen. CD4 T cells are very high in the lymph nodes and a little lower in the spleen. And because PET imaging is inherently quantitative, we can see that. And this is very nice, and we want to use these fragments to image the different immune cell subsets. However, while zirconium-89 gives these really nice high contrast images, the long half-life of zirconium does not allow for repeat imaging. So you would have to wait 10 half-lives, which is a month later, to be able to image this mouse again. So in order to do immune profiling or repeat imaging of immune cell subsets, we would need to use F18, circling back to what Anna talked about, that the cis diabody is the ideal fragment in combination with fluorine 18. And so I want to present this work about the anti-human CD20 cis diabody labeled with F18 for same-day ImmunoPET imaging that we have published in the European Journal of Nuclear Medicine and Molecular Imaging in 2019. Again, like Anna introduced, the diabody has peak uptake around 1 to 2 hours and a half-life of 2 to 5 hours, and that's a perfect match for fluorine 18 imaging, and F18 is an almost ideal positron emitter, 97% positrons, and then it has a very short half-life, so that would allow for repeat imaging. The antibody fragment used in this work is based on a therapeutic antibody obinutuzumab that was developed by Glyco and Roche. So this is a humanized glycoengineered type 2 CD20 binder, which means it does not internalize at the same rate that, for example, rituximab does. And I have reformatted this into a 50 kilodalton cis diabody that is biologically inert because it doesn't have an FC region. And then because this fragment now targets human CD20 to be used in preclinical models, we use a transgenic mouse model that expresses human CD20 on the mature B cells. And then later on, I will show work where we use the syngeneic mouse B cell lymphoma that has been transduced to also express human CD20, so we can detect this B cell lymphoma in the context of normal CD20 expression. And so for the F18 radiolabeling, the easiest method to do that is to use SFB, which randomly labels to lysines, and that's a fast and easy reaction. However, because these fragments are very small, the chance that you conjugate somewhere in the binding site and interrupt antigen binding is quite high. So we wanted to compare the SFB labeled diabody to a site specifically labeled construct. And here we used the C-terminal cysteines that were reduced using TSAP. And then we can use a F18 prosthetic group that contains a maleimide for site specific conjugation to the thiols. So here's how the first one, the FB anti-CD20 cis diabody looks like in the human CD20 transgenic mice. You can see early on uptake in the spleen and you see blood pool, and then the diabody clears to the kidneys. And at around 1 hour, you can see excretion with the urine in the bladder. And then the lower images are wild type mice, just to show you that this signal is CD20 specific. And in a mouse that does not express human CD20, there's no background. The site specifically labeled diabody looks very similar at early time points. You see targeting in the spleen, you see blood pool and then clearance to the kidneys. But then around 1 hour post-injection, we start seeing activity in the GI tract and some secondary clearance through the gallbladder, which then leads to a lot of background signal in the abdomen. So these are the same images scaled up just to show you that we can see the lymph nodes using the F18 labeled cis diabody and the spleen, and both tracers specifically target the CD20 B cell containing tissues. However, the radiochemistry does impact the excretion and we get a lot of activity in the GI tract. But we think that the FB labeled anti-CD20 diabody is a good tracer for same-day imaging. So these images were taken 4 hours post-injection, and this would allow us to then image the same mouse with a different F18 labeled tracer the next day. And here we used FDG to do that. And these are the same human CD20 transgenic mice, just to show you the background that FDG has in these mice. So you see the hard kidneys, bladder, and uptake in the Hederian glands. So we then use this system of repeat imaging with FDG and anti-CD20 cis diabody for serial PET imaging. And this is now the human CD20 transgenic mice that grow disseminated mouse B-cell lymphoma that homes and forms tumor nodules in the liver. You can see that here with the FDG imaging. And then we image the same mouse the next day for CD20, and you can see the same tumor nodule in the liver showing up. But then on top of that, you now see the systemic expression of CD20 in B cells in the spleen and also the lymph nodes. By day 19, this B-cell lymphomas has overgrown the entire liver and the FDG signal gets quite diffuse. And then the next day, again, we image with anti-CD20 where you get very nice specific signal in the liver. And then again, you see the spleen and the lymph nodes, the normal tissue expression of CD20. And I'm just showing this because the resolution of PET is not always so good that we can see individual tumor nodules in the liver or very small structures. But I just want to point out that even in the limited disease at early time points, we do see increased uptake in the liver, and that is before we see a difference in mass in the liver from tumor growing. So even if you're not able to distinguish a specific metastasis in the liver, the overall uptake is higher. And so that is why we think that the F18 labeled anti-CD20 cis diabody would also be able to image, for example, tumor infiltrating B cells. And so to sum this up, we have this set of cis diabodies that specifically target immune cell subsets, CD4 and CD8 T cells, CD20 B cells. And we have used these diabodies successfully to monitor the localization and expansion of immune cell subsets, both in the tumor and systemic. And because diabodies have the very fast renal clearance, we can image at early time points, and that enables us to use these diabodies in combination with F18 in order to be able to conduct same-day imaging and repeat imaging of these different subsets and get an idea about the immune profile. And now to show you an example of clinical applications, I want to talk about the first in-human study using a radio-labeled anti-CD8 minibody to image tumor-infiltrating T cells. This was the featured article of the month in May in the Journal of Nuclear Medicine. And in this study, a anti-human CD8 radio-labeled minibody was used, prevmirlimab berdoxam, and that minibody is based on the mouse anti-CD8 antibody OCT8. It was humanized, it was reformatted into the 80 kilodalton minibody format. And again, it's biologically inert because it doesn't have Fc effector functions. So this phase 1 study was supposed to show that we can use this minibody to detect whole body and tumor CD8 leukocytes. It was recruiting patients with metastatic solid tumors. And there were 2 phases, a dose escalation and a dose expansion phase, but all patients got the same amount of radioactivity, 3 millicurie, and then serial PET scans at different time points to investigate biodistribution, the dosimetry, and uptake of this tracer. And so here are the patient characteristics. In total, 15 patients were enrolled in this study, 6 patients in the initial dose escalation phase, where patients got different masses of minibody. The second phase included an additional 9 patients for dose expansion in 2 dose groups, 0.5 milligram and 1.5 milligram. Of these patients, the majority had melanoma or non-small cell lung carcinoma. 8 patients were on immunotherapy. 2 patients were on targeted therapy. 2 patients had discontinued treatment, and 3 were treatment naive. And overall, the injections were safe and well tolerated, and there were no drug-related adverse events. The serum clearance of these radio tracers was by exponential and depended on the injected mass of minibody. At very low mass, the clearance was very fast, which is probably due to antigen-mediated clearance. And then for the dose expansion cohorts at 0.5 and 1.5 milligram, here the calculated fast and slow half-lives based on radioactivity or based on the functional minibody measured by ELISA. The whole body PET imaging showed that the tracer shows CD8 specific uptake. So the first and most prominent tissue you can see is the spleen that contains a lot of CD8 T cells. But then you can also see bone marrow and lymph nodes throughout the body. And then there's CD8 specific uptake in the tumor tissue. Here's a metastasis in the neck area that is most prominent around 24 to 48 hours. And then for the normal tissue uptake, you can see the liver, pretty stable activity in the liver. And then later on, you see some activity in the GI tract, which confirms hepatobiliary clearance. You can also see some uptake in the kidneys. And here's that semi-quantified, the CD8 specific uptake, again, highest in the spleen, both for the 0.5 and the 1.5 milligram cohorts, and then a specific uptake in the bone marrow and in the lymph nodes. For the normal tissues, the highest uptake is in the liver and in the kidney. And then the normal tissues such as lung and muscle have very low background activity. The tumor uptake was variable, but was seen in 10 out of 15 patients, and that included 7 of 8 patients on immunotherapy, 1 patient that had discontinued therapy, 1 patient that was on targeted therapy, and 1 of the 3 treatment-naive patients. And although this study was not designed to correlate tumor uptake with response to therapy, for 3 patients, there's clinical follow-up available. And I want to show you these patients in more detail. So the first patient is a metastatic melanoma, and this patient was on immunotherapy with pembrolizumab. And this is an FDG PET scan after 8 months of therapy, where you can see 2 avid lesions in the neck area. The same patient then got the CD8 PET CT scan about a month later, and the same nodules in the neck area show very high CD8 signal, suggesting that CD8 T cells are infiltrating these metastases, and also that maybe some of the FDG signal is due to CD8 leukocytes and not tumor cells. And then the follow-up imaging of this patient showed stable disease. So that would support the hypothesis that the high CD8 signal suggests an anti-tumor immune response. The second patient with the locally advanced melanoma, here's the baseline FDG PET scan, where you can see 2 metastases in the left axilla. And then after starting 28 days after starting treatment with pembrolizumab, the same patient gets the CD8 PET CT scan, and these lesions are very hot for CD8, suggesting a high number of tumor infiltrating CD8 T cells. The follow-up image at 3 months after treatment showed a complete response, and that has lasted for more than 2 years now. The third patient had a liver carcinoma. Here you can see the primary tumor and then some regional lymph node metastases. 2 weeks after starting treatment with nivolumab, this patient received a CD8 PET CT scan, and you can see that the primary tumors in the liver show very high CD8 signal. And then here are the follow-up scans 2 months and 3 years after treatment. You can see that the nodal metastases have resolved and that the primary tumor in the liver is much smaller. So this partial response has lasted more than 3 years now. So in summary, we can use CD8 PET imaging. It is safe and well tolerated, and it allows us to visualize the whole body biodistribution of CD8 positive leukocytes, both in the tumor and systemic. And that may allow a prediction of early response to immunotherapies. And so I want to end with the next steps, so what's going on currently. So the phase 2 study using this tracer is ongoing. And for this phase 2 study, patients will receive a pre-treatment CD8 PET scan and biopsy, and then an on-treatment CD8 PET scan and biopsy, with the goal of correlating the CD8 PET signal with CD8 IHC, and also correlating these images with outcome in these patients. And with that, I want to thank my colleagues at City of Hope, especially my mentor, Anna Wu, and all current lab members, the people working in the Small Animal Imaging Center at City of Hope, and then all my former colleagues and mentors at UCLA and the Imaging Core at UCLA. And I want to thank you for your attention, and hopefully we can answer some questions.
Thank you so much, Kirstin, for a wonderful presentation. And it's fantastic to see the correlation between the CD8 images in patients and their clinical responses. So now the floor is open for any questions. Please feel free to type them in the chat for either of the speakers, Kirstin and Anna. So I have a specific question related to Kirstin, your work on the site-specific labeling. And you showed that with the site-specific labeling, there were some higher signals in the clearance organs. But of course, there's certainly some advantages having site-specific labeling for these antibodies. So could you comment on the advantages and disadvantages of using one versus the other?
Sure. I mean, of course, the idea was to use site-specific, so we would have more control over location and stoichiometry of the radio label, right? And we wouldn't impact the antibody binding. For the CD20 diabody, it looks very similar at early time points. So if you're imaging even earlier at 1 or 2 hours, it might not make that much of a difference. And then later on, some of the metabolized tracer gets reabsorbed and clears through the GI tract, and that gives you all that background. But if you're faster than I am and you image at 2 hours and not wait that long, it might still be the better tracer. We haven't given up on the site-specific F18 labeling. I mean, the SFB labeling is just a lot easier, and we're trying to translate that for the other CD4 and CD8 diabodies. But if that impacts binding, we might go back to the site-specific version.
Great, thank you so much. So we actually have a couple questions. I'll go one by one. And this may be for both speakers. What do you think are the primary challenges facing the application of clinical ImmunoPET through the radio-labeled mini-bodies?
Primary challenges for clinical application. I think we're still at a point where we are pretty sure that it works, but now we need to show that the information you get is useful. Right now it can be applied clinically, but it's not FDA approved, it's not broadly available yet, so it's still within the confines of clinical trials. But I think that the main, so we can do these clinical trials with the clinical agents. It's not cheap at this point, because I think when you scale up, then you get economies of scale, and one hopes to get to that point where it would be a reasonable value, for what you're getting. But I think really the key is to continue these studies to understand whether or not this approach actually provides useful and actionable information, and then we'll really get broader clinical use.
Thank you to both speakers. Just being curious on the role of radiometal chelator ligands effects on the antibody.
Okay, so for the diabodies we use with zirconium, with the radiometal, we do use the site-specific conjugation methods to make sure that that is away from the binding site. Other than that, we have not seen that adding a chelator to the antibody fragments changes their targeting or in vivo behavior.
I think that's one of the beauties of the antibodies, because they actually are fairly large proteins, so that adding a chelator and radiometal has less of an impact than it would, say, on a peptide or a small molecule, where you do have to pay more attention to the overall changes in charge or potentially hydrophilicity or hydrophobicity that can impact the clearance. So it's less of an issue with antibodies. I almost say it's not an issue. We pretty much see very similar behavior, say, with antibodies and fragments labeled with different chelators and radiometals.
How do you envision the use of these ImmunoPET tracers to look at specific B cell and T cell subsets?
Well, so ideally, we would want to use F18 labeled diabodies where we can, in a short period of time, look at B cells and T cells in the same location, in the tumor or in the draining lymph nodes. What we do not know is what we are really looking for, right? Is it just co-localization of B cells and T cells? Is it ratios of CD8 versus CD4 T cells? Or what will be the best biomarker to early on know if a patient is going to respond to immunotherapy or not?
So actually, we have quite a bit of questions. I didn't realize there's some in the Q&A as well, so let me jump straight to those. I truly appreciate the fact that the development of humanized antibodies and antibody fragments had a significant impact on the development of these agents. Thank you for the comprehensive overview. Fantastic presentations given by the speakers. Kirstin, in that patient with positive CD20 nodules, would you have classified the tumor as progressing based solely on the FDG PET?
Oh, so that was CD8 specific ImmunoPET in the patients. And I think I do not know what these patients were diagnosed as, if that was progressing just by the FDG-PET. I think the FDG-PET often also shows CD8 T cell activity if they're infiltrating, right? So it's not just the tumor. I do not know how those would have been early on.
Yeah,
the FDG-PET scan was early, right?
Yeah, I think in the field, there actually has been a concerted effort to come up with standardized, modified RECIST criteria for looking at patients in immunotherapy protocols because of the fact that you get this confounding of signal. So you'll often see more lesions. So they look at things, how are there new lesions appearing, or are lesions disappearing over time? So it's not just a yes, no anymore if you're looking at immunotherapy, because people realize you can get a mixed signal. I don't know what that would have been called because I don't know enough about what else was seen in the patient. But it is the field, people doing immunotherapy and doing FDG-PET have come up with modified ways to look at how things are changing to try to, at least with FDG-PET, have a better understanding, of what's happening in a patient.
Have you tested the stability of F18 cis diabodies in serum and at different pH in any of the medium?
No, we have not done extensive stability studies with the F18 labeled diabody yet.
Is site-specific labeling impacting the structure and consequently biological activity of the cis diabody?
Well, it could, because we are like reducing the cis terminal disulfide that stabilize the diabody. So after conjugation, we make sure by size exclusion that the diabody structure is still intact. And then of course, we also test immunoreactivity to make sure that the biological function is maintained.
So I'll try to see if we can get through all these questions. It seems like everyone's quite excited about ImmunoPET, of course. Thank you so much for the great talks. Being tumor immune cell interactions so dynamic and heterogeneous, how can PET imaging schedules be defined so that we are sure to collect most of all essential biological information? Repeated imaging would definitely help, but could you comment on the feasibility for translation of PET-CT in the same patient for multiple days through the course of therapy?
I would say that this really is a new modality and a new type of imaging. So we're still learning, what is going to be the best time, say during the treatment course to image, and when repeat imaging might be useful. So, it's hard to say now, how we're going to define imaging schedules, feasibility of repeat imaging. In clinical trials, you can set studies up, to actually look at, repeat imaging. For example, in the phase 1, there was serial imaging after a single injection, and now in the phase 2, there's 2 injections. So I would say it's really early still in the process of learning how to use these agents and what is the most useful time point. Because I think actually the $54,000 question is, do any of these images predict ultimate outcome in these patients? So I think it's still an open question. I really can't answer it directly. It is feasible to do repeat imaging, but with the zirconium, as Kirstin mentioned, you can't re-inject and re-image until you wait, as in the phase 2, we wait 4 or 5 weeks for the original activity to decay so that you can do a fresh injection and a fresh image. So that is the limitation right now. We hope eventually F18 labeled agents will move into the clinic, and then we could do repeat imaging, on a shorter schedule. And the question is, will it be repeat CD8 or will it be CD8 and a different biomarker? Those are all possibilities. They can all be explored in clinical trial. That part, it's feasible. And then what ends up being useful and in broader use, it really just depends on what we learn. And again, where the most interesting and useful information is going to come up. I hope that answers the question.
Can you foresee the potential for dual tracer ImmunoPET using different targets as potential modalities?
We're counting on Craig Levin to help us pull out different PET radionuclides. There are a lot of people using different approaches to try to image more than one biomarker at a time. either serial F18, you know, like a day apart, as Kirstin has mentioned, or one right after the other, but doing dynamic imaging and modeling so you can subtract, you know, the signal from the first probe from the second, or co-injection with PET radionuclides with 2 different half-lives like F18 and zirconium and pull them out at different times, or next generation scanners that can take advantage of the unique signatures of some of these PET radionuclides, for example, gammas in coincidence, to try to pull out the different signals from 2 different you know, radio biomarkers labeled with 2 different PET radionuclides. So we would love it because biology is not simple, you know, and we have a wonderful gift in FDG. It's so useful, but you know, when you get to try to get a little more specific, we ideally could look at more than 1 or 2 biomarkers, you know, at about the same time to see what's going on. So I think there's high potential, and certainly there's a lot of research going on in this area.
Thanks for the impressive presentations. Do you think there could be a potential for cocktail imaging using, for example, FDG and zirconia 89C8?
Yeah, I think we just touched on that. You know,
Yeah.
you maybe inject both, and or how would you do this? Because you don't want to drown out. It have to be a lot of F18 FDG and a little bit of zirconium if you wanted the early image to really reflect the FDG, or you'd want to do the FDG first and then do your image at 2 hours and then inject the zirconium and have the patient come back the next day, you know, or a fancy PET scanner that can pull apart those signals. Because there is you know, there is additional gamma from the zirconium that you might be able to use to distinguish the 2. So all good questions.
And I think I've finally exhausted all of them. And we're kind of close on time, but so for sure, I'd like to invite everyone to join us at the social lounge. It's on the left-hand side of your screen. Just click there and it'll take you to another session where the speakers will be present for casual interactions, any particular questions you want to follow up on, or any questions on general mentorship and menteeship. And I guess what I wanted to end with is a question to the both of you on your thoughts on mentorship and being a mentee as well.
So I will just really quickly, because we're at the top of the hour, I've tried to set a good example. You know, I care about the work I do and that it's done right. And so part of mentoring, I think, is setting a good example, showing your interest and excitement for the work that you're doing, and expecting other people to come to that standard. I feel my role often, though, is to provide resources and get out of the way of smart people and to solve problems, protect them from problems. I think it's important to understand, you know, of your mentees, what their goals are. Do they want an academic career? Do they want a job in industry? You know, what their goals are. What do they want? Do they want to learn new skills? You know, what are their goals? But I'm actually going to toss the ball to Kirstin and ask her, what makes a good mentor?
Well, yeah, so I think what you said is right, because you provide the space for growth, and I have to say what was most motivating for me, not only the example or the work that you do, but also that you, I think, never ever said no to any idea or project or experiment I wanted to try or do. So there was never any restrictions in what I could come up with ideas to explore. That was always supported and allowed in the lab for even the weirdest things to at least give them a go. Before you understand yourself that it was like not a good idea.
I got that from Sam.
Yeah.
He would never say no. He was always, what if? Go for it.
Thank you so much. It's very important for us to have great mentors, but also to learn from our mentees as well. And with that, thank you so much to our speakers, Dr. Anna Wu and Dr. Kirstin Zettlitz from the City of Hope. And we look forward to seeing you at the social lounge. Thank you so much, everybody.
Thank you.
Thank you. See you over there.